Hi, I had a question about the oxygen hemoglobin dissociation curve, and so I wanted to try to help out with that. So, to put this into context, if you take a look at the notes at page 11, this is where I start talking about the oxygen hemoglobin dissociation curve. So, basically, what we're talking about here, the topic here, is the transport of oxygen. In other words, once oxygen enters into the blood, let's say, during external respiration, or exits the blood during internal respiration or unloads from the blood into the tissue, how is it traveling in the blood? Where is oxygen in the blood? That's what the transport of oxygen discussion is all about. And so as you can see here, and as you know already from our last unit, the majority of oxygen travels in the blood bound to hemoglobin. 98.5% is bound to hemoglobin, and the remainder 1.5% is just dissolved in the plasma. And so what I do in the notes right away is I start talking about the factors that are associated with um, the strength of this bond between oxygen and hemoglobin. And then, what we do in this class a lot is we look at that in a graph form. So that's basically what I'm doing here. But as far as the oxygen hemoglobin dissociation curve, let me try to show you some pictures and explain this a little bit better. You might recall that hemoglobin is the molecule that fills red blood cells, right? So red blood cells have over 250 million molecules of hemoglobin per cell, and each molecule of hemoglobin is made up of 4 protein chains, and each of those protein chains contains something called a heme group. And if you take a close look at the heme group, what you have is an organic ring surrounding an atom of iron. And when oxygen goes into the blood, what it does is 98.5% of it, we're saying, binds to hemoglobin, and it binds right to that iron atom, which means that one hemoglobin molecule could transport up to 4 molecules of oxygen. And so what the oxygen hemoglobin dissociation curve does is it just looks at this dynamic. So let's take a look at that. So taking a look at the oxygen hemoglobin dissociation curve, I can see on the x-axis that we're looking at the partial pressure of oxygen. Okay, so the partial pressure of oxygen from 0 to 100, and they're even showing me partial pressure at the lungs of oxygen is about 100 millimeters of mercury, and partial pressure of oxygen at the tissue is about 40 millimeters of mercury. And then on the y-axis, what they're showing me is the percent saturation of hemoglobin. So how much hemoglobin is bound to oxygen. So basically, when hemoglobin binds to oxygen, you might expect, and we don't see this, so you might expect that as the oxygen, the partial pressure of oxygen goes up, then the percent saturation of hemoglobin goes up. Something like this, like a linear function. You know what I mean? So that as the pressure goes up, saturation goes up, and you see this linear function, and that's not what we see. Instead, what we see is this kind of S-shaped curve. And so what this is showing us is that basically, as the partial pressure of oxygen goes up, once you get, say, I'm just going to make some, random marks on a graph here. Once I get 1 oxygen to bind, it structurally changes the hemoglobin molecule so that binding the second one is even easier, and binding the third one is even easier, and binding the fourth one is easiest kind of idea. That's where you're getting this kind of S-shaped curve. And so when you talk about the percent saturation of hemoglobin, what you're actually talking about is how many molecules of hemoglobin within the red blood cell are saturated with oxygen. Because of this dynamic, binding of 1 oxygen makes it easier for the binding of the next, making it easier for the binding of the next. Hemoglobin in the red blood cell, it's either going to exist as oxygenated or not oxygenated, deoxygenated. And so if hemoglobin is oxygenated, people call it oxyhemoglobin, and when it's not bound to oxygen, it's called deoxyhemoglobin. And so to read this graph, what we could do is say, at a partial pressure of 100, we know that the partial pressure inside the alveoli of oxygen, it equals 100. What how much hemoglobin is saturated. And I can see it's almost, 100% saturated. It's a little bit, under that, like 97, 98% saturated, that hemoglobin molecules are saturated leaving the lungs. Or in other words, there's 98% oxyhemoglobin and 2% deoxyhemoglobin within the cell. So what about at the tissue? So at a tissue, once this oxygenated blood goes through a capillary bed at a working tissue, the partial pressure of oxygen at the tissue, they're telling me, is about 40. And so if I take a look at this graph, what that's showing me is that oxygen will leave hemoglobin, but hemoglobin remains about 75% saturated. So even after the hemoglobin, even after the blood leaves the tissue, the deoxygenated blood, so to speak, the deoxygenated blood still has 75% oxyhemoglobin and 25% deoxyhemoglobin. So that's how this is read. I hope that helps. So in the notes where I'm talking about factors that affect the bond between oxygen and hemoglobin, basically what I'm doing, just looking at the notes again, is I'm saying, okay, well, you have to have a partial pressure of oxygen gradient in order for it to move anywhere. So that gradient is going to be the driving force for the movement of oxygen either onto or off of hemoglobin. There's another factor, it's a waste product, it's called 2,3-DPG, and that waste product is from like the metabolic activity of cells, and what it does is it enhances the release of oxygen. So young healthy red blood cells will have a lot of this 2,3-DPG, it's going to enhance release of oxygen. This is actually going to, slow down as the cell ages, and this is a factor that the Red Cross looks at in determining how long they can store blood, because you don't want to give blood to someone that doesn't release the oxygen, which it gets, worse at releasing the oxygen once it gets older. So those are two very general factors. But then the other factors, I grouped them together because I thought this might help you remember that these are indicators of an active tissue, like an increased partial pressure of carbon dioxide. So when we're working, we're generating more CO2, and that CO2 is going to weaken that bond. Also, as you're working, the pH decreases. Just think of like lactic acid buildup in a muscle, and that weakens the bond. And also in working muscle, we start to get hot, right? It generates heat. And so an increased temperature will weaken that bond. And you can look at that in association with this curve too. So for instance, taking a look at temperature, here's the oxygen hemoglobin dissociation curve again. Let's make a comparison. So I think this is 10, this is in increments of 10, so there's 10, 20, 30, let's say here's 40 millimeters of mercury. So this is what it's like at the tissue, and normal body temperature is that purple line. And so what this is showing me is just what we were looking at before, that at normal body temperature, that the hemoglobin is about 75% saturated with oxygen. But look what happens if our temperature is higher, like the bright fuchsia line. If our temperature is higher, it looks like at that working tissue, the hemoglobin is only going to be, say, like 55% saturated. So do you see the difference? It let go of more hemoglobin. That increased temperature weakened the bond between oxygen and hemoglobin. Or let's make another comparison. Let's say, it's colder. And so here at temperature of 20 degrees Celsius, much colder, I can see that the hemoglobin remains much more saturated with oxygen, right? Over 90% saturated at that colder temperature. And so I talk about this in the notes, and this is what I mean by a curve shift to the right or to the left. And so if we take this normal body temperature, this is the standard curve, you can see that when you're looking at a factor like increased temperature that weakens the bond, how that curve is shifting to the right. And if you take a look at a factor that increases the strength of the bond, what will happen is that curve will shift to the left. Okay, so if it was colder, I can see instead of being the standard curve, it has like a more steep slope to it. So going back to the notes, I talk about those things here. Okay, so the unloading or loading of the first oxygen facilitates unloading or loading of the next. That's something that you can see on the graph. Creates that S-shaped curve. I explained that. The curve shifting, I explained that. And I also give you a nice graph here, which you don't have to guesstimate like I was doing on the graph. I'm giving you a table so you can see if you had a partial pressure of X, what is the percent saturation of hemoglobin? So that's right in the table. And I would definitely recommend watching this video. If you watch this video on the oxygen hemoglobin dissociation curve, I think you'll feel much better about this topic. So I hope this helps. Thanks.